Hello and welcome to our animation showing RNA transcription. We are going to cover the different types of polymerase, promoters, transcription, splicing and toxins. There is a lot of molecules responsible for this complex process and are shown here. This is Bertie and he is going to introduce briefly what RNA transcription is.
This is a transcription bubble and it shows how the strands unwind and allows NTPs to be added. There is a coding and a template strand. The NTPs are added in the 5 to 3' direction.
Now, we are going to look at types of RNA polymerase. There are 3 types of polymerase. We are focusing on RNA polymerase 2 as it transcribes mRNA. Promoters are key to starting the process of transcription and they help to activate transcription factors to recruit RNA polymerase. In prokaryotes, the sigma subunit of RNA polymerase recognizes the consensus sequence. This is a sequence taken from nucleotides in different genes that are similar or identical. To allow RNAP binding in prokaryotes, there must be a TTGACAT sequence at -35 and a TATAAT sequence at -10. Upstream of this, the sigma factor dissociates when transcription is initiated. In eukaryotes, a core promoter of 4 elements are shown in purple, red, green and blue. There is also an upstream promoter element. Within 20% of all eukaryotes, the common sequence is present. A very important core promoter is the Tata box. It is present 25 to 35 base pairs before transcription starts and it indicates which strand needs to be read. RNA polymerase 2 requires a set of proteins called transcription factors and these help position the polymerase, unwind the 2 strands of DNA and release RNA polymerase.
Hello again, I am going to show how NTPs fold and how they bind together. In RNA transcription, uracil and adenine bind together, cytosine and guanine also bind together. They are bonded via hydrogen bonds. There are 2 bonds between U and A and 3 bonds between G and C. When these proteins link into beta sheets or alpha helix structures, phosphodiester bonds are formed. This occurs when the sugar backbones of the bases link together to form a chain.
Transcription results in the formation of mRNA. This contains introns which are non-coding sequences and exons which are coding sequences. At the splice site, phosphodiester bonds are broken. A spliceosome will remove the non-coding regions and will leave the functional mRNA behind ready to be translated.
Drugs and toxins affect RNA transcription. Some of these can be found in plants and fungi. These can be extracted and used by the pharmaceutical company. In fungi from the Amanita family, a drug called alpha-amanitin is found. It is a cyclic peptide made from 8 amino acids, and it inhibits RNA polymerase 2 and 3, disrupting RNA transcription. In RNA polymerase 2, it has 12 subunits and alpha-amanitin degrades subunit 1 by binding near the active site of the polymerase. During transcription, a trigger loop is formed. This loop is a polymorphous component which promotes nucleotide addition by making direct contact with the NTP. This trigger loop can be targeted by inhibitors to block the addition of NTPs. Alpha-amanitin is chemically linked to an antibody-targeted amanitin conjugate. Tumor cells rely on transcription for the expression of oncogenes that suppress apoptosis. By pausing or stopping the transcription in cancer cells, oncogenes are transcribed less frequently so the growth is reduced. Thank you for listening to me. I hope you enjoyed our animation.